in the cloud. All right. There we go. All right. Well, today I'm actually kind of nervous because this is like a lot of stuff that I don't actually all understand it yet, but it's coming together. It's really exciting what I do understand, and consolidating everything that I understand so far and sharing it with you is also exciting and a joy for me. So thank you for your interest in compost tea and for following this research or this journey with me. I'm trying to share my screen. There we go. You all see everything? So good to see you. So I'm going to, okay. I'm going to, I consider this like my thesis dissertation. So this is a very like formal compared to what I normally give. But I hope that it's also understandable and that you'll see some of the benefits or where you might want to use or just relax a little bit. That's my hope, is that everybody coming off from this webinar will be able to like relax a little bit on like how to use it, whether or not they should use it, or especially with all the fanfare that's been made on compost tea. If you've been going deeper and deeper into your farm or your garden, and so that's where I'm at. I'm Nikki from Permaculture Gardens, and I compost in 3 ways. I compost using bokashi. I compost using and I'd ask everyone to mute. I hope that people could mute if they could come in. Okay, thank you. I compost using vermicompost, worms mostly, and let's see, I can mute there. I compost in a slow compost and a thermophilic compost, actually 4 ways. And here's a video. Oops. Let me know if you can hear it.
We can't hear it.
Oh, sorry, you can't hear it. Let me try that again. I have to stop share and reshare with the sound. Okay, here we go. Thank you for that feedback. Of this, some of these beds have already been harvested, so I'll have to account for that. But it's not lost upon me how fortunate, how privileged I am that this is my daily job, that I get to do this every day and look at the plants and see how they change and study them. So I'm going to go ahead and take measurements. This is a control. This is the untreated, nitrogen treated bed, and it has been harvested the most because these were, believe it or not, these were huge. And this is the compost bed. Beyond there, so each of these rows have different N treated, control, and compost treated. So this would be a compost bed right here. This is a compost bed. This is a control, and that is a N treated. This is a control, a compost bed, and an N treated. And from viewing this, it's not quite sure. You can see the different stages that the plants are in. So we're going to tally everything up and see whether just by weight alone and crown diameter we can find whether or not there are measurements to say that one treatment is better than the other. Aside from that, everything else here has been the same. Some weeds are proliferating, a few, very, very, very few. But for the most part, it is very much a controlled growing environment. Okay, so to clarify, N-treated is nitrogen treated. So that would have been, the comparison was, oops, I'm sorry, let me get out of there. The comparison was nitrogen or conventional type of growing versus, let me see, how do I get out of here? Versus compost tea treated versus control. So where I didn't do anything to it. So those are the three beds that the three kinds of treatments that I have. So when I say N treated, it was the recommended, like given what the soil was, I tested it, the recommendation for putting on nitrogen in it. And in particular, when you grow leafy greens, this particular study is only on leafy greens. And so that is what was recommended, addition of nitrogen every 2 weeks. And then they had a measurement that was used, if you wanted to go deeper into it, that's in my research. So today I wanted to talk about, well, what compost tea is, or where we found our definition of compost tea, problem versus solution, why use aerated compost tea in the first place, and then how I went about my research and did it, and next steps, meaning subsequent researches on this topic that we're exploring, subsequent questions. So first off, compost tea, what is it and what is it not? So Google says at first, I mean, the thing that comes to mind is compost tea is something where you put compost in water and you spread it. And that's anything that, the term tea kind of brings to mind, like you steep it in water and then you use it. Just like we steep a tea bag in water and we take it, right? And so, but when I've looked into this, and when I Googled whether or not compost tea was good, the first thing that came up was it has no real benefits. And so I wanted to explore this, because how is that possible that there are no real benefits, but so many people have said that this is really something that works? So I looked into our research specifically on aerated compost teas, and that is where our definition is evolving. This particular research was done in Korea, and it was done with leafy greens, again, lettuce, soybeans. Well, sweet corn is not leafy green, but the lettuce there kind of pulled my attention. Let me just see here, who else needs to be muted? There. And in their study, there was a significant difference. Of course, the kind of compost that they also used was different. There was a significant difference when they used compost tea in greater concentration. So the more compost tea they used, the more concentrated it was, the bigger their soybean grew. And the more diverse as well the compost tea was. So in this case, they had different compost. They had a rice straw compost, they had a medicinal herbs compost, and they had a vermicompost. And when they put them all together, those particular treatments grew the best. So it's great. And you can see this in this chart, where when it was most diverse, they had a greater greater bacterial growth in their compost tea with the more diverse mix. So the more diverse your compost is to begin with, the more diverse your tea will be. So what is the problem? Well, why my problem was really coming not so much the greater problem is a lot of people use pesticides. That's the bigger picture. And I was trying to find a solution for farmers that would lower their input and yet and increase their output without having to buy, even if they want it to be organic, buying organic amendments is expensive. So is there one thing that would possibly be the solution to that, to their inputs, of lowering their input? And would this possibly be it, right? So that was my bigger picture, was can we make it cheaper for organic farmers to grow this so that we don't use pesticides? Can we make it more compelling for conventional farmers to not use pesticides if we can prove that compost tea is a viable option, right? Is the same, it performs in the same way. So of course I'm rooting for compost tea in the beginning, but trying not to let it cloud, of course, my research. So the title of my research is Effects of Aerated Compost Tea on Swiss chard, kale, and lettuce production in Virginia. So specific to the South. And this is a grant that runs until March of next year. And our first our first definitions of compost tea were really indebted and grateful to Dr. Elaine Ingham, because she was the one really, I feel like, popularized this in the last 10 years. And before then, and it came from when she writes, you can look at her compost tea manual, which is free online. What she writes there is, people have been composting since the Egyptians. And yet somebody had come to her and had a big composting brew machine or whatnot. And since she was a microbiologist, she was really interested in learning whether it really increased the tea, and so the way that she does this is, as she and her students do this, is they take a sample under a microscope and they count manually the number of bacteria in a drop of compost tea or compost. So when you send it to her labs, that is what they do. And so, so through the course of this research, I have also been sending it to her lab, certified lab. The closest one to me is in North Carolina, and she says ACT, which is aerated compost tea, should be brewed for 24 to 36 hours to allow sufficient time for aerobic microorganisms to multiply to beneficial levels. The brew should be used immediately after brewing to ensure the highest microbial activity. Okay, so aside from her, there are very few researchers really that definitive papers, and this one, since she was working with USDA, it really has a widespread impact on organic growers, especially these statements. And so a part of this research is also going back to those and retesting them also with the latest technology that we have now. So what are the types of compost tea? And Elaine Ingham's and her students have certain nomenclatures for calling the different teas such. And honestly, I found it really confusing because only her and her students will call something aerated compost tea. And then now it's grown to activated aerated compost tea. When she first used the term aerated compost tea, it was a given that you would feed molasses or some sort of a feed to the compost tea. So you wouldn't just put a bag of compost in water, you would also put some sort of a bacterial food in it that they could survive in. And for her as a microbiologist, I can understand, like, you want to proliferate, you want to amplify their number, and that's why you do that. But then it wasn't, people were taking that the wrong way. Some people were feeding, some people were not, especially since in the USDA, for organic growers, they have a very specific standard. They have a national board that says, organics board that says, oh, you cannot put feeds at all in your compost tea because you will make the pathogens proliferate. And that's their definitive statement. I'm like, so how can I be an organic grower if I have to not go by the standard every time? If Elaine Ingham says you need to feed it, and then you guys say that you can't, you're not supposed to feed it. So that was already for me confusing from the get-go. And then this term activated or aerated compost tea came about because of that. So some people were feeding, some people were not. And so activated meant to say you fed it. Compost extraction later on, when people wouldn't feed their teas, the word extraction is what Elaine would use for saying that something was not called a tea if it is not bubbled at all. It needs to be aerated to be called a tea. And so I felt like this is even more confusing. So for the purpose of this talk, everything that I've done is aerated compost tea that I've fed in this research. But if people call it compost tea and it's an extraction, as we shall see, there are benefits to so many other ways of compost. And finally, we have the leachate, which is the dregs of the compost. So if you're in a bin system and you have a compost bin and you have the yucky stuff that perhaps is more anaerobic or low oxygen, those are the dregs. And they're, from my experience of the writings of Elaine Ingham and her students, these are the least favored because of their tendency to be pathogenic and aerobic. And so I wanted to explore all this, is it really, etc., all that she says. And so why aerobic was the first question I asked. So this is her statement. Compost and compost tea need to be produced in oxygen-rich
aerobic environments. This ensures that the finished product is rich in nutrients and beneficial organisms. When compost or compost tea is made in anaerobic conditions, nutrients are lost to the atmosphere, which is why anaerobic compost stinks. The end product of anaerobic decomposition may also contain alcohol, phenols, organic acids, and proteins that are toxic to plant tissues. And rightly so. So when you smell, and she says this in her manual, when you smell and it's stinky, you know it's bad. So we don't want it because it's releasing nitrogen that should be in plant soluble form. It no longer is. So it's like evaporated into the air and you can smell it in the ammonia. And that's what we don't want our compost or compost teas to smell like at all. So if we look at what we're trying to do in the compost tea, soil, as I've talked about in the past, is made up of three things, right? The chemical components, the physical components, and the biological components. And this particular talk on compost tea is, of course, targeted to cultivating the bacterial components or the microbiological components. Bacteria is not the only thing in compost tea, but for this study, I went mostly on the bacteria. So there are different kinds of bacteria. You have aerobic bacteria in the spectrum of bacteria, facultative bacteria, that means they can straddle both worlds, and anaerobic bacteria. And honestly, most of the pathogens are found in the anaerobic, which is why I think that bubbling it, if you're going to spread this gospel widespread, you want to make sure that nobody cultivates the anaerobic bacteria. But the harm in that is that it might be looked at as black and white, that only bubbled tea is. I'm now thinking of bubble tea, like that the high schoolers like to drink. Only bubbled or brewed compost tea is the way to go. So but we're finding that there are all of these bacteria that I've listed here on the screen in front of you are beneficial to plant growth, to plants, okay? And you can see the different, I've put like the benefits to plant. What do they, disease suppression, nitrogen fixation, produces antibiotics, and they live and overlap in function across these different spectra. Some of them are aerobic, some of them are low oxygen, some of them are anaerobic. And again, the human pathogens that we are familiar with, Enterobacter and Clostridia, are found in the anaerobic. And that's not to say there aren't any, or maybe there are, no, we haven't found it. But that's just to show you that there are good stuff also in anaerobic bacteria, which is why bokashi, which is a fermentation process, works. And this is why, as we will see here, answer this question, is leachate bad? This was the dregs of a worm bin that my collaborator, Dr. Zach Jones, thank you, Zach, if you're watching, and any farmers are watching out there, Zach Jones and Agrego Data is the company that was very thorough and is very good about helping me analyze my data. So if you need DNA testing, farmers especially, for your soil, vermicompost, that's his specialty. He's the place to go. And so this is the dregs of his neighbor's compost, and he took a sample of the bacteria here and he put it not under the microscope like Elaine did, but using DNA testing, which is now, a more up-to-date way of figuring out who is in your worm bin, or who are the players in the bacteria. And this is fun because the way that this works is you put the worm juice or whatever, you put a drop of it in a vial that sort of flash freezes. This is my layman's term, so forgive me. Flash freezes the bacteria. So it's like whoever's in there is in there, and we will find out who they are, and they cannot move or multiply anymore. And then they send it to the lab and they DNA extract it, and they figure out who exactly was in that tea. And what he found out was that this was pathogen free. First of all, this is a vermicompost bin. And I wanted to also share that there are studies that show that when you pass compost through the gut of the Eisenia fetida worm, which is the red wriggler worm, let's use that worm over here, that's used in vermicompost thing, that there is sort of like a layer of filtration that they neutralize, or they lower the pathogen load, as it was done in this research. It was intentionally given that, like, it was intentionally inoculated with pathogens, and then they were passed through the gut of the Yersinia fetida, and I believe that they were lowered. I don't think they were totally eliminated. Maybe, they didn't eat everything, but there was a significant load that it was very, it was like they clean the bacteria for you. They clean it from the pathogens. And so this dregs, it was pathogen free, one. Second, it was more diverse than Compost Tea was. And Zach does this for a living, where he compares Compost Tea with vermicompost. But it was less diverse than the vermicompost itself. And the players that lived here were the microaerobic bacteria, the low oxygen bacteria, and facultative anaerobes that were helpful to plants. So PGPR stands for plant growth promoting rhizobia, the kind of bacteria, plant growth promoting bacteria that helped in chewing our nutrients so that they can be uptake by the plants can uptake them, and also they were found to be disease suppressing bacteria. So this carbon, he noted, is more soluble and fermented and more useful to plants than they would be if they were not in this vermicompost. So he suggested if you pass this through and combine it with biochar, which has a lot of airspace, and these plants are they can live in both the anaerobic and the aerobic places, then they can fill out those airspaces and be even more effective and multiply and helpful for your plants. So does it need to be aerobic? Conclusion: ACT is particularly effective when freshly applied to soil or plants, providing a surge of, and here is what we're going to talk about next, a few families of bacteria, of mostly air-loving and more pathogen-free beneficial bacteria. However, non-aerated teas or compost extracts might be more suitable for applications where a slower release of more diverse microbial benefits is desired over time. I know that's a mouthful. So why do I say a surge of a few families of air-loving and more pathogenic bacteria? Well, we'll get to that in a little bit. But first, another question that I wanted to ask along the way here, and this is actually a question that came as a result of me doing the first research, was, well, how long am I supposed to brew this for? Because Elaine Ingham says 36 hours. That's also a different, from what I knew, this is Elaine's, from Elaine's user manual. Dr. Ingham's user Compost Tea Brewer's manual. So you see that she has the activity spike at about 24 hours, and that's when, for this particular brew, and they're using up the oxygen and then they die out. So there's a bell curve for bacteria. And in this, the study, the Korean study I was basing my first study on, they said, oops, let me just turn that off one second. What do you see? When did they spike? When was their peak microbial population? So these are the days: 0 day, 1 day, 2 day, 3 day, 4 day. And then these are the units of their population density. It's very small. Okay, so we are showing here a spike of bacteria on the 3rd day. So then it's variable, sometimes from what we know. Let me see here. And this is SARE, Sustainable Agriculture Research Education. Shout out to SARE for making this particular training possible and my grant possible. So why am I going here? We went from here. I wanted to ask this question too. How long does it take for the bacteria to grow, and what happens to them while they are in their growing phase? So let me just make sure. Yep. Welcome, Miriam. All right, so the second research that I'm doing currently, is a comparison of the microbiome population induced by different microbial foods for the production of a safe and effective aerated compost tea. So again, I'm big on the safe because I want to make sure I'm within USDA guidelines whenever I'm brewing these things, and that when I teach these to farmers, they feel comfortable that they are growing within USDA guidelines. So we already, what we know so far to answer the question, how long should we brew, is that if it's warm, your biology, the microbes in your tea will be more active and will spike sooner. So we looked at two bell curves of one spiking at 24 hours and one spiking at 36 hours. It could be 6 hours, 12 hours, 24 hours. We don't know. This is just from what we've been told by other scientists and other brewers. If it's cold, it will take your microbes longer to reach the peak state. So I wanted to answer this question in the second study as well. So this is where I'm going to stop share, but maybe you can see my screen as I switch tabs here to this chronograph. So this is a chronograph. A chronograph is how how my bacterial data is represented right now when through DNA testing. So like I mentioned, I have also sent the same samples to a lab of Dr. Elaine Ingham's, where the microbiologist or the microscopist will look underneath and count manually how many I have and give me a diversity of them. And what has happened the past 2 times that I've sent is that all my bacteria is dead by the time that it gets to them, even though I rush ship it, that the vermicompost is always alive and thriving and within the limits recommended, very, very robust biology, but the tea is always like useless, no biology, no bacteria. So we know that bell curve is correct, that it dies eventually, and it dies within transit to them. But the DNA testing can tell me who was there at the time of my harvesting. And so, and this is data from the first research that I did, where I sent it to the lab and I had them sample my vermicompost. So this is what my vermicompost looks like. You can see how many different families are there, and some of them are not even, if they look like this, RCPG, they're not really studied yet. they're phylum. So they're very diverse, right? And then when I go and when I sampled and harvested at I think this was the 24th to 36th hour, what do you think happened? And you can reply in the chat. As you can see, I feel so uncomfortable with not engaging anyone and just presenting. So this is the vermicompost, these PEG 2 and PEG 3. So there are both vermicompost tea samples. So they were harvested at 24 to 24 hours, I think. And PEG 1 is the vermicompost itself, the compost before it was added to the water, and this was after it was added to the water and brewed for 24 hours. So if each of the colors represents a different bacterial family, what happened after 24 hours to those bacteria? And feel free to unmute if you are having a hard time.
Well, it looks like the bacteria just die,
Yeah. So that what
most of
happens is,
them.
yes. So what happens is they don't, that's where I'm still testing it, right? I'm not sure if they all die, like how much of them died. So yes, so the diversity, these guys, all the other colors died, but then the red ones and just a few of the other colors dominated, right? And that I think I was only the second. I don't know if I'm only the second, but I've only been, Zach Jones, Dr. Jones has just been doing this a few times, and this is already so, I kind of like, not well known in the field of agriculture, that after you brew the tea, we're thinking we're amplifying everybody. Like all of this is what we're thinking we're doing. We're amplifying all of their population. When in fact, we may only be amplifying a few of them. But is this necessarily good or bad? So that's where, I mean, you continue the conversation. Like, so now that you know that.
So Nikki, can I ask a question?
Yeah, go for it.
So looking at that data, I mean, I guess there's a couple of ways or conclusions you could draw. Either that the compost tea doesn't ever produce as much bacteria, or that it's dying off before they are able to test it to identify it.
Yeah, so that one, I know they're dying off before they test it in the microscope, but they're, because of the DNA technology, like I can capture them and flash freeze them in the vial right as I harvest them. So then everybody's DNA is there and flash frozen. Nobody can reproduce. Nobody can die anymore. I mean, they're dead. So that tells me who was totally in there, and that for me is like the fascinating thing. I think this is actually, I'm looking at this slide, I'm like, this is not my data. So he was not as diverse. Like this is Troy Hinkie. This particular vermicompost was not as diverse as mine. This was the other sequences, because when I made this talk before, I had to use other people's data because mine didn't come in yet. And so this one, but you can still see there was a drop. Like there's proliferation of members of only 3. So I was trying to find out. So what's happening? And so from my chronograph, right? What do I feed them now? So okay, there's a drop in the drop from 24 hours to I'm amplifying only a select few. Is this good? Like, am I supposed to be targeting just these few families, and because they are plant growth promoting, or are they disease suppressant? And if so, then maybe I'm just I should only use my compost tea for disease suppression, right? I shouldn't bother so much about it at the beginning of my growing season, especially since they're not diverse bacteria. So from a permaculture standpoint, I'm like, well, diversity is key. And the less diverse it is, then, I mean, the less desirable it is, unless you have a specific purpose for selecting for those bacteria. So what you see here is my labeling list of the different feeds in my second research, where I wanted to see, okay, so what should I be feeding them? Because then there was a whole other debate on never feed your compost tea molasses, because that'll proliferate the pathogens, And it's true, like when we're not supposed to eat too much sugar, and it does spike, the bacteria. So I decided to test it and see who exactly was spiking at what point in time. And so I had a control. I had plain alfalfa meal, plain kelp meal to feed, plain humic acids, and plain molasses, and fish hydrolysate. So these are my like singles. And then I did combinations of the two to see how they would react. And I had 32 replicates because I had 16 samples done at 12 hours. And then I did it again at 24 hours and tried to see what happened to the populations over that course of time. So what do we see in these results? So first, the vermicompost has no detected pathogens again. So I think that those studies that the vermicompost, running it through the gut of an Eisenia fetida, definitely lowers the pathogen load for your, lowers the pathogen load of your compost. So first of all, I tried not to put manure in this. I mean, this is not a manure compost bin, right? This is just kitchen scraps, shredded paper, vegetables, no chicken manure, no anything. So that way I tried to make sure that it was not pathogen, it was pathogen free. Most of the compost teas agriculturally that are being made are made in farms, of course, that have the manure as one of the ingredients of their compost. So if they were to use that, then maybe passing it to the vermicompost would lower the pathogenic load, and then they could make the teas out of them. So that's kind of like what I think this is, the testing is showing. But more testing is necessary, because that's not particular. My study is for creating a pathogen-free and effective tea using what feeds, right? Making sure that I'm using the right feeds, because they were saying in the National Organic Boards, don't feed anything at all. And then Elaine Ingham later on amended her own writings to say, don't feed molasses, because that's what will proliferate a pathogen, not knowing what you're starting with. But if you know if you're starting with vermicompost, then you feel a little bit more confident that you have a pathogen-free base. So vermicompost has no detected pathogens. There's nothing in the tea that wasn't first there in the compost. So all these players here that are amplified or living came from this. And there's nothing that appears outside of what you originally had. If we don't feed the tea anything, so one thing that Elaine Ingham would say is, if you don't feed the tea anything, then it's a compost extract, then it's not a tea, right? It's not useful. But we noticed that if you don't feed the tea anything, that was my control, after 24 hours, the bacteria were still alive and diverse. So it was with nothing, just bubbling. And of course, like, this is coming from a specific, I cannot say that you would be able to replicate whatever I did in terms of vermicompost, because whatever, my kitchen scraps are different from yours. And so, that's just my specific disclaimer. Molasses, fish hydrolysate, and kelp meal all tend to decrease bacterial diversity after 24 hours. And different feeds select for the abundance of different bacteria. So what exactly those bacteria do, I'm not yet there. I'm still going to be chatting with Zach Jones about what each of these bacteria are doing, and why that is. There's one bacteria, he said, that comes up only after 24 hours, and that's the flu something. It's not even listed here. Yeah. All right. So do we want to select for specific bacteria is the question. What do you all think? Anyone?
Maybe if you have a particular problem, but in general, it seems that it just makes it more complex or complicated.
Yeah, that's what I wanted to say, like, from a permaculture standpoint, and also reading this book called by John Sitka called A Soil Owner's Manual. It seems to me that we don't want to have like this reductionist point of view when we're growing, right, in agriculture. We want to be as diverse as possible. Diversity is stability. And unless what I'm trying to do is deselect for bacteria. So I'm deselecting pathogens. That's what I'm trying to rule out. Like, I want to grow this bacteria, but I don't want any of the pathogens in. And I think that one is probably a good reason to deselect, for I'm not growing those particular bacteria. Or maybe if a bacteria later on is something that we need, but even if it was, that kind of mentality of I need to, I would like to, we do that with fungi, right? I would like to inoculate mycorrhizal fungi, but mycorrhizal fungi doesn't work with brassicas, so you want the specific fungi that you want to inoculate with. So it is kind of, I think you have to know what you want. And to your point, Vivian, it could be useful for some time in the future, when we know a little bit more about what we're doing. But I think more variety is better, or diversity. But in general, I think for the gardener, for the average gardener, especially if we don't have to do all that work, can you imagine like all this brewing, and you feel confident. You should feel confident if you already have a vermicompost bin, or if you have compost and you just put it with water and water your plants with it. That can be enough, and that can be just as good as if you brewed it. But I still want to answer those questions. Like, what exactly are we doing, and how did this come about? Why is everybody adopting compost tea aeration then, and in large scale for large places? And people swear by it. You know, some people will have a brew, especially vermicompost is so powerful. I just came from California a few months ago, and when I was there, there's this company called Boogie Brew, and they were trying, the permaculture people that I was with got their made their compost teas with them, and showed like the difference between leaf growth before and after the compost tea. And it's like, wow, fascinated by this. And they said, the best stuff for this, for the compost tea, like soil fertility applications, comes from California because of the marijuana growers. They're so like meticulous about how they're brewing it and what's in it. And for the most part, what was in it is vermicompost again. And if they had a feed for it, it would be the exoskeleton, which I did not test, the exoskeletons of mealworms. So it would be frass, mealworm frass, because those were, and this goes to another place. I'm only doing bacteria in this research. I'm not doing fungi, primarily because the lab that I was working with did not test for fungi at this time, but now they will. So my future researchers will also test for fungi, and maybe we can see, when you add fungi to that mix, it opens up even more questions, a bigger can of worms. So still pending analysis for the 12 and 24-hour tests, and I still am lacking bionutrient data because the research purpose, again, of my initial research was, first, is ACT a viable alternative to synthetic fertilizers in organic farming practices? To assess the growth performance, root development, leaf quality with and without ACT compared to nitrogen treated and control groups. Nitrogen, because as I mentioned earlier, that was the conventional application that was recommended. And Miriam, you have a good question. Do specific plants need specific bacteria? That is a great question. That's what I'm trying to also find out. I don't know. I think we know that they know, and from that same conference to another research, another person who was exploring research on plants was talking about something called endophytes or endophytic bacteria and fungi. And these particular bacteria are carried in the seed coat of seeds. And when seeds that you would harvest, not sterilized seeds, and it's almost like, I explained this to Rachel in our past check-in, it's almost like the inoculation of immunity that a baby, when they pass through a birth canal, has because of her mom's mucus. The seed coat has microbiome that is necessary for the immunity of the plant that will germinate from that particular seed. And so that those endophytes, those endophytes are not universally present in all seeds, even though they should be, right? So this lab, Dr. James White in Rutgers University, has done research on it so that they took out all the endophytes from a seed, they sterilized the seed and saw how it grew, and then they also did it so that what happens if you inoculate it with, put back the endophytes into the seed, and can you do that? So this inoculation later on, I'm not sure, it's very novel, like in just the last 10 years as well. So I don't know to what extent that's being done. I don't know if I can do it, and that's my third research proposal, which I don't know if I'm going to get because I haven't applied for it yet. But if I were to ask another question, I'm looking at the context in which I grow, and it's Northern Virginia wineries that do not do organic at all. Like there's only one certified organic winery in Virginia and only 3 in the Northeast, and all of them spray. And so the reason that I want to explore this is to figure out if you can inoculate it later on, because rootstocks may not have the endophytes, grapes, that is, may not have the endophytes. And so that's a good question that just went on to my next research. Thank you, Miriam. Number 3, to evaluate the impact of ACT on nutrient density in leafy greens. Aha. So now we're going to look at what exactly I weighed out. And this is where, is ACT equal to or greater than an N fertilizer, at least in leafy greens? Now, that's my question. Does ACT make leafy greens go bigger? Or does it make it grow better, nutrient dense? So now, again, I'm not the best grower out there. So this is my compost tea beds that you saw at the beginning of this presentation. They're all laid out. Everybody was planted with Swiss chard, kale, and lettuce, but the treatment to each bed was different. So the beds at the bottom here, 7, 8, 9, were the ones that always were the first growers and the first ones planted that I got to plant. And but in the end, I was able to harvest from all of them last spring kale and lettuce, and not all of them had Swiss chard, so I felt like I needed to do that whole Swiss chard thing. And I tried to weigh their crown, measure their diameter and weigh them. And this is the result of that. So I want to preface this by saying that because I am a farmer more than I am a scientist, I started this research with soil that was already, I would think, rich, because I couldn't bear to grow in clay dirt. And I was told, as I'm learning, I should have started with dirt clay instead of mushroom compost. So everybody had mushroom compost, the control, the N-treated, and the compost. And like I said, this particular study looked more at the bacteria than at the fungi, because they could not DNA sequence the fungi for me at that time yet, only the bacteria. So I don't know what role the fungi had played in this, especially since the leafy greens, kale, I don't know what their associations are with fungi. So control gave me the most lettuce, but closely followed by compost, and the N-treated was the least. So that was surprising to me. My lettuce, I've shown these graphs before in the group, the GIY community. My lettuce was sent to Bionutrient Institute to test for polyphenols. So they test the richness of the polyphenols in my lettuce, and that mine fall here in the 20th scale in general. And the Brix rating was actually above average, but now that I actually have a refractometer and I'm using it, I'm like, this is pretty low. Maybe it was taken in the early morning. So the Brix measures sucrose in your leaf, and sucrose is a complex sugar. So it would have had to have good bacteria, good nutrients, good growing basis before it can produce sugars like sucrose. So my lettuce was growing, this is the Compost Tea lettuce, was growing at the higher than average of all these samples. And if you're wondering where all these samples came from, these are like farmers and samples from Whole Foods, samples from conventionally grown store that have been fielded to the Bionutrient Institute, which is a research institute, private research institute. And antioxidants, it was also measured. So my lettuce did not have as many antioxidants as the mean did. Now looking at the kale, the control again had the best kale, and the ACT was similar to the nitrogen, but just a little bit greater than the nitrogen in terms of weight. So I feel like I wanted to redo this, but I'm redoing it on soil that's already been used and has, the mushroom compost is decomposing in there. And so I feel like the control will always outweigh. I don't know, maybe you can also see that if you grow in mushroom compost and you have leafy greens, you don't even have to do anything to it, and it'll just perform really well if you leave it alone. So leaf grow is the way to go, at least from my data here. And this is just 9 plots, so it's not for everybody, but this is what I'm finding. Finally, I also looked at the root diameter, and the largest diameter it was found in ACT. Plants treated with ACT showed significantly larger average root diameters, which could indicate more robust root development. And the smallest root diameters were in nitrogen. And I'm still analyzing why this is so. Is this because the nitrogen treated plants don't have to work so hard to create roots because the nitrogen is right there available for them? I don't know. And that's where, and then control diameter, moderate root diameter, control root that showed moderate root growth. So I think that after this, I also want to look at, well, is it really more nutrient dense if I grow, is it really more nutrient dense if I grow with aerated compost tea than with not? Or does aerated compost tea not have an effect because it doesn't have the diversity of bacteria? And should I be using just plain compost tea instead, you know? So all these questions, can you say more about your mushroom compost? It's just, you have it delivered, you ask for mushroom compost in the soil. Like for us, it's Loudon Milling, I think. And in Sterling, it's Virginia ground covers. And instead of having them haul you topsoil, you say, oh, I want leaf grow. And leaf grow is the same as mushroom compost. So it's leaves that have been decomposed by mushrooms. Okay, so that's what I mean by mushroom compost. Finally, I bought a bricks meter, which you can buy off of Amazon, like $20, $25. And they won't say to test the sweetness of your plants. It'll be like to test for honey or viticulture or something of that sort. So, but I'm using it to test for the sweetness in the leaf of my leafy greens. So when I looked at the different treatments, I hope that you can hear me. It says my internet connection is unstable. I saw that, of course, the control reading at 1 p.m. was the lowest. The untreated was the highest, which surprised me by one reading. And then ACT treated was always almost the same as untreated. So I looked at different times, 1 p.m., 7 a.m., and 5 p.m. And the ACT treated was always like one behind the untreated. And again, my Swiss chard sample falls here. It's above the mean of brick samples sampled by bionutrient. So in that spectrum, that is. So it's pretty sweet. Any questions so far? So, this is the slide where I'm supposed to say my conclusion. I guess my conclusion is, if you went back to that question, is aerated ACT just greater than or equal to nitrogen fertilizer? I would say yes, it's comparable too, because you almost have the same bricks reading, right? If you have it, is as a fertilizer. Does it grow a bigger plant? Yes, for lettuce in my plots, but I don't think necessarily. So I don't know yet. I'm still going to plant some more. So I still have a bed that's waiting to be harvested. So beds that are still waiting to be harvested. In general, lettuce, yes, it was greater. For kale, it was a little bit greater. So it does it grow bigger? The biggest was control. And then the third question was, does it grow a better crop, a more nutrient dense crop? And right now we're seeing that, compared to the N-treated, it was not. It was comparable, but not the same, not as high as the N-treated. And this was also replicated in my lettuce. For the N-treated, it was 4. And for the ACT-treated, it was 4.4. So it was like super close comparison. Okay, question. Thanks so much for sharing. I have to leave a little early, but I appreciate your research. Oh, you're welcome. All right. So now we go to how to make aerated compost tea. In particular, so aerated compost tea is oxygenated. I have a fish tank. A fish tank bubbler pump. And then you want to make sure you're using living organisms, so worm castings or a compost mix. And then food for the life, like I mentioned, fish emulsion, liquid kelp, humic acids is what I used for this. And so the next step is a comparison of the microbiome population induced by the different microbial foods. That's what I just shared with you. And actually, that's it. That's it for now. That's all I have to share. Do you have any questions? That is I have all these comfrey growing. Is it late to make the tea? No, in fact, I have a video here of Dave making the tea, because this is like the practical part. And here, let's see. I'm gonna stop share, and then I'm gonna share with sound just to make sure that I share properly. Share with sound. Yep. Okay.
Hi, this is Dave from Permaculture Gardens. We do a lot of experimenting, and sometimes these things don't work out, but we thought we'd start sharing with you the experiments as we're doing them, just in case they do turn out to be useful, and it just guides you through kind of the things that we're experimenting with. Here we're trying to make a non-aerated compost tea. So I've gathered about 1 kilogram or 2 pounds of a mix of comfrey and stinging nettle. And this is supposed to be a really good mid-season drench or nutrient feed for things like tomatoes, peppers, your fruiting crops in the summer that need that extra boost. So I've gathered all this. I've already started chopping it up. So all I've done is I'm going to wrap it up. I have here some Agribon row cover. It's like a breathable plastic row cover that I use in the winter for protecting my low tunnels. But I'm repurposing it because if you have something like an old stocking that's supposed to also, like a hose, you could also fill all the materials in there. You have to be a little careful with the stinging nettle. It actually will sting you. That's why I'm wearing gloves. The reason why I'm chopping this up instead of just wrapping it all up is that you want to increase the surface area so we can extract as many of the nutrients out of these plants as possible. So after I cut all of this up, I'm going to wrap it up like a bag, and then we're just going to put it in a non-chlorinated water for about a week. We'll stir it every day. It's non-aerated, so I'm not using a pump. So this is probably one of the easiest compost teas that you can make. Now, this is the first time I've ever made this, but once I've made it, we're going to test it on some of our tomato plants and see if it boosts growth, and then we'll report the results to our tribe. Okay, so I've cut this up. All of it in there, and then I'm going to wrap it up.
You want my stockings?
Well, you don't have old stockings, right?
Well, there's I have stockings. Yeah, I do.
Well, I've already got this all set up. Go with this for now. Because I think if you already have some Agribon, it's probably easier just to repurpose that. Okay, so we got it. So now we'll just soak it in here.
It's just cold water.
Okay, so it's just cold water that I got out of a rain barrel. If you're going to get it out of your tap, let it sit for 24 hours so the chlorine can dissipate before you put the plant materials in. And that's all you have to do. So we're going to let this soak for the next week, and then we'll use it as a foliar spray and a root drip for our tomato plants and our peppers. Thanks.
All right, and that was it. 2 pounds in how much water? In 5 gallons. Yeah, totally. And it doesn't have to be like with stinging nettle. It's just if you have it, or any other leafy green you want to put in there. Any other questions before I end this workshop? Since it's at the top of the hour. Was that helpful at all? Does that make you feel better about using or not using compost tea?
So do you recommend aerating it between 12 and 24 hours, or how long for permaculture, tea?
Yeah, so Like I said, I was still, so if it's cold weather, it takes a longer time, they say, for the biology
Right.
to develop. But I just stick with 24 hours because then it's also easy for me to like, you have to be practical too. And I'm like, okay, I'm going to start this at 5 today, and then I'm going to come back at 5 tomorrow, to stop, turn it off and use it.
Okay.
Yeah, but good question. Where are you growing?
Northern California, where I first saw you.
Oh,
So, yeah, so being I don't care so much about being strictly USDA organic. So, should I add some molasses, like a tablespoon in a five
yeah.
gallon bucket?
Yeah, you can.
Would that be
I
more helpful?
so what we're finding is molasses decreases the diversity a little bit, but it spikes it up really quickly. So if you add molasses, you can harvest it there's no difference between 12 and 24 hours. That's what we're finding so far, at least from my testing. So you could even harvest it earlier, sooner if you wanted to, using molasses.
And I've been taught to foliar spray, which is
Yeah,
better,
yeah,
house plants, everything,
yes,
rather
yes.
than giving it, just some water in the ground. And then when do you add, so you talked about a little bit about the biochar and so how would you apply that into the ground with the biochar and the tea?
Yes, so foliar sprays are specifically for the plant leaves,
Yeah,
so that
leaves, right,
yes,
right. I
because
know.
they
So you mix,
yeah, but the
so
biochar would be a drench, a root drench. So you can apply it in two ways: as a foliar spray to spray it, or as a
uh
root drench.
okay, that's a compost tea, but the biochar needs to go in the ground, right?
Yes, yeah, the biochar needs to go in the ground. So you would sprinkle the biochar first in the ground and then drench it with the compost tea.
Okay, so you'd put how much biochar around the plant? Whatever's recommended, or do you know?
Yes, so if you buy the biochar like
Right.
Wakefield, then they will have a recommendation in the back of the bag. Some people make the
Uh-huh.
biochar, and then I don't know. I just follow whatever's in the bag, and normally I just kind of sprinkle it around to make sure
Uh-huh.
that yeah, I don't have a specific 1 cup
So you're
per bed.
okay. Normal practice is you take some biochar and you sprinkle it around the plant after the plant is growing,
Yes, go
and then you put some.
even before you could do it even before.
Yeah, you can add it to the soil. Okay, okay, and then but you typically just put it around the plant is what I'm asking you. Your practice, and then
Yes.
you add the compost tea. At what time in the grow?
So for this research, I've been adding it every 2 weeks from seed germination. Actually, I even
Uh-huh.
tested so it's not good as a seed germinating
Uh-huh.
medium, but it's really
Uh-huh.
good once it's a seedling onto when you harvest it. We're still
Uh-huh.
spraying with compost tea. So that every 2 weeks was my practice, just because I was basing off of what the Korean research was doing. And
Uh-huh.
some people like the tomatoes, for instance. So this is leafy greens, but let's say tomatoes, they will really
Mhm.
lay it on there in July, when they're really, I mean, you guys still have peppers, right? So I don't know yet, but you're putting it on there while it's growing and it's producing fruit so that it doesn't flag the production. But I would suggest like just try it when what are you growing?
Collards right now.
Oh,
So
collards, yeah.
collards and cabbage.
Perfect.
But I have,
So for
yeah,
every week actually for me, I was doing every week.
okay.
Yeah. And sometimes, so this is where the testing becomes useful, is you may need to do that more initially, like just to get the biological flywheel going,
Okay.
but you may not need to do it later on in the season once you see that it's super fertile and really performing. But
Uh-huh.
initially, you'll have to do a lot of, like now, I'm looking at this bed and I'm like, I need to hurry up this bed because need it to grow faster. And I'm like,
Uh-huh.
every other day I'm putting something on there if I'm out there anyway, not just water, but something with compost as well.
Last question. Have you had problems with pill bugs getting into your vermiculture and using the vermicompost in your
Yeah.
tea? Are you spraying out the eggs of the pill bugs?
No, well, so put the vermicompost in a mesh and then
Uh-huh.
I suspend the
Bubble it.
mesh, yeah,
Okay.
into the water. So
Right.
that mesh is like I squeeze out all the juice that I can while I'm bubbling it, and then I use that. Pill bug wise, so I don't have pill bugs in my vermicompost, but I do have mealworms and I have red wrigglers. And I wanted it just to be a red wriggler worm, and all sorts of flies. Flies went in there and everything, and maybe I have pill bugs as well. So what I'm doing is, I guess if I did have a problem that was pill bugs and specifically, I would separate out, okay, the vermicompost compost, maybe you'll be somewhere else so that if you know pill bugs really attack your leafy greens, and then just
Huh?
use the juice of it if that's an issue. And at different times of the year, like do you ever get a season where the pill bugs are dead or they don't, it doesn't get cold enough there for them to die?
I've not had so much of a problem on on my greens. I'm just talking about I don't want to make compost tea and add pill bug eggs
Yes.
to my tea because they're in there. I've been
Yeah. So I just
killing
say
it off with some
filter.
GE.
Yeah.
So just squeeze it out. Just
Just
squeeze the
squeeze,
bag.
yeah, so squeeze that mesh bag out, like wear gloves, squeeze it
Yeah.
out. You want to squeeze out as much as possible. And then you're just getting the juice and the bacteria of, and it's still suspended there and it's brewing for 12 or 24 hours. And then you just discard the bag or put it somewhere else or use it in your
So don't
compost
put the
bin.
bag in. Don't put the bag into the aeration mix. Just squeeze out the juice and then bubble
No,
up the juice.
put the bag in
Put the
there,
bag in.
but the eggs shouldn't pass through the mesh holes. Sorry if
We
this is,
hope not.
let me see if I have a video of me actually with the bag. More information is here on my actual website where
Uh-huh.
I have, let me share the screen and you can see what
Thank you.
yes, because maybe that's where the confusion is coming from is there's no visual of what I was that I was doing. So thank you for that. Permaculturegarden.org, Compost Tea research. Okay, so there's my 5 gallon, and then where is the bag? Okay, here we go. So here in my picture, you see how I have this mesh bag? I bought that
Uh-huh.
off of, yeah, Tea Lab. So any nutmeg milk or milk bag, they sometimes call this like a nut milk bag. It's
Uh-huh.
fine mesh. So you can squeeze out the stuff, but the compost for the most part stays in that bag and it's suspended here. So while it's brewing, it gets bubbled and jostled and the stuff comes out of it into the brew. And then if pill bugs are an issue, don't put that back into your garden. You can put this compost back into your compost bin,
Uh-huh.
and then they can do their work there and be more productive, but they won't necessarily go directly into your garden. Does that help?
Oh, you mean what's left over in the bag, you mean?
Yes, what's left over in the bag, but keep that bag in there the whole brewing time.
Oh yeah, of course. But you have to squeeze that bag before you start the
Yes. Yeah, you have
that's
to squeeze.
standard? That's standard?
Yes, that's standard. And on the website, you'll see like a how to actually do the compost thing. Sign up. Okay, wait, hang on a second. I have how to make compost tea somewhere below here. If you just
I'll
where
find it.
is
Thank you
it?
very much.
Yes, how to brew.
Thank
And
you.
then this one, you're welcome. Yeah, then this one is a link to Troy Hinkie giving these lessons and answering all those questions. But you're welcome. I'm so glad you had so
Thank
many
you very
questions.
much. Thank you.
Vivian, thank you for the time and effort. Sometimes the more you know, the more questions you have, and that's wonderful. Yes. Oh, Sandra, thank you. My pleasure. Well, welcome and everyone again to this world of compost tea, and I will present again next year about this as I learn to grow, maybe more often if people are more interested in this. But I'll always try to update it on this website, on my website, permaculturegardens.org. There is a link to my compost tea research just on the homepage, and anything I find, I'll try to update this, and you'll see the final researches here. So thank you for your time. And with that, I will let you all go. And here's to more fertile grounds now that we know the benefits of compost and compost tea. Bye.
Bye.